Well, we started with a discussion of the molecular basis of DNA. In our last couple of lectures, we talked about the basic building block, these units that make up DNA. And of course, that's a nucleotide. We now know that nucleotides are made up of a phosphate group. Then they're made up of a 5-carbon sugar, a deoxyribose sugar, and also then these bases, which are either single-ringed pyrimidines or double-ringed purine rings that make up ATGC. So now we want to talk about how do we put all that together? What's the physical structure? If we have those building blocks, we have these forms that we can put together, how can they work? And as we do that, they're really people that were thinking through this. There were two basic critical questions, and of course, the first one is that physically, how do you construct these units together to give us a model that is some molecular form? We know that DNA is continuous with chromosomal type structures, so how does that link together? How do we put these building blocks together to make that form? The second was any type of genomic material, whatever it is, okay, in our case, DNA, what are the biological properties? In other words, how does that structure actually work to make the biological properties of that DNA work? And this could be true for anything. It's basically the question is this: what's a successful genetic material? On this planet, DNA, primarily some RNA, is our genetic material. If we were to go to another planet somewhere in the future and we find out maybe it's a sulfur-based unit instead of a carbon-based genome, well, still, what's going to have to be there to make it work? Okay, and so some of the things that had to go, and so this had to be answered, right? One of the questions is we have to know how to put it together. The second question is, okay, now that you got it together, how does it actually work as a biological agent? So what's the first thing? Well, obviously you need to be able to transfer information from generation to generation, and you need for that information to be able to be read to make phenotype. Obviously, so whatever you have, it's got to be stable enough you can get it passed in some form, and then you've got to get a pull apart or looked at so that you can use it as an effective blueprint. Then it must be able to store lots and lots and lots and lots of information. And this fairly small package, it's got to have a lot of code that will work to it. So that's one of the things you have to answer. How's that going to work? How can we store huge amounts of information? Think about it. You basically have about the same amount of DNA as a sequoia or a whale or something else, yet we're totally different creatures. So somehow that information had to be stored there. Different information had to be stored there and function. So information storage was critical. And then, of course, you have to be able to replicate it. And take that information that you have, that starting stuff, and make new stuff. Develop the embryo and then to a fetus, into an adult tree or to a human or whatever. Has to be able to copy it and replicate it. Once you get the adult made, you've got to be able to replicate again for various things like skin cells and things. So you've got to be able to do that with some fidelity, as it's called. So you've got to be able to replicate, keep that DNA and the message that you're trying to send there correct over time. And then obviously when you have reproduction, you've got to replicate again so you can pass the information on faithfully to the next generation. And then finally, while you got all that going on, you need to be able to change it from time to time, okay? Successful genetic material at time X in a million years may not be the most successful type. You need some way to adapt. We always have environmental changes going on. Things heat up, things cool off, the amount of water that's available goes up and down, the food sources change, so you have to be able to adapt to those changes. The way we do that and pass it on to our progeny is through mutation. Now, you don't want too much mutation, you don't want just going crazy, you want it to be, but you want to have a variability there so that the good things can be used in the future, right? So again, any successful material has to have this. So people were thinking about these two big problems. What does it all look like? And then how do you put it all together to make it work in a biological sense? Now, starting with the first part, with this idea of how it's all put together, we'll look at Irwin Chargaff. Irwin Chargaff did lots of things over a long period of time, but during this period, about 1944 to 52, he was able to really extract some really fundamental information. What he was looking at, he was able to isolate some good quality DNA from a number of organisms, and he described the various ratios of ATGC in DNA in these organisms. Now, he was doing this for several reasons. One, just to figure out what was there, but also he was trying to test what was known as the tetranucleotide hypothesis. This been a hypothesis that had been the kind of the mainstay of thought about DNA structure throughout the 1900s. And it had originated with Levine in about 1910. Now, you remember from our last lectures, we talked about Levine was about 1909. He described the major components of RNA. About 1929, major components of DNA. Well, he had proposed this idea of what's known as a tetranucleotide hypothesis, the idea that A, T, G, and C formed the corners of some kind of structure, and that they were each level was an A, T, G, C in sort of equal molar concentrations. And then in his view, then proteins were somehow incorporated into that. Remember, we have chromatin, which is a mix of DNA and proteins. He thought the chromatin part, the part that makes up the proteins, was the part where the message was stored. Well, of course, by the 1950s, we know this is no longer true. It's actually the DNA itself that's coding the messages. But he proposed, and this was basically the idea, there was some kind of regular form that these structures took. And so you can imagine, and DNA was serving in his mind as some kind of superstructure, kind of like the Eiffel Tower here, kind of an exposed superstructure. And with this 4-base model, if you were to stand underneath and look up, you would, underneath this long stretch of DNA, you would see something that looks similar to this. That would be A, T, G, C, and this would just keep repeating as we go up along the lines. And his basic idea was that these phosphodiester bonds would connect up between these sugars in some regular pattern like this, okay? So that we would have a phosphodiester bond here, 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 and here between A, T, G, and C. And then there'd be some kind of covalent other bonds that would then do the next layer, and you'd have another A, T, G, C as formed, okay? So that you would have, again, these very regular types of structures, if you, what you could see if you would cross-section our DNA. Now, this is the hypothesis. The hypothesis is that DNA is made up of these very regular structure with all of the units equal. This leads to a very straightforward prediction. The prediction, which is what, we test predictions in science. The prediction is that the molar quantities of A, T, G, and C should be approximately equal. So this is what Chargaff was trying to do. He was trying to test this prediction. He was saying, okay, if I can extract DNA from a number of organisms, I would predict, if it's going to be this correct Levine model, that ATGC will be about equal. So he was able to pull DNA from things like viruses, and it's relatively close. Again, this is fairly crude DNA, fairly crude model, so you can't expect it to be exactly perfect, but he was seeing approximately 25% A, 25% G, C, roughly equal. So about a ratio of about 1, okay? When he looked at E. coli, okay, the bacteria, he saw, well, it's a little different, but it's still right at the range of that. It's just, again, within experimental error, same. Fungi, pretty much the same. Again, he's seeing differences, but they don't appear to be that great. He's starting to still see ratios around 1 of all of these. And then he starts to hit things that are multicellular, truly multicellular, such as flies and other things. And lo and behold, whoa, the system really starts to fall apart. There's no longer 25% A and close to 25% G and C. We're seeing 30% A and only 20% G and things. So somehow we're really starting to see odd things. But interestingly, the amount of A to G and T to C are still around 1. Okay. However, the ATGC ratios are now getting way off balance. And what he's really seeing is that the amount of A is approximately equal to the amount of T, and G is equal to the amount of C. When he gets to humans, lo and behold, very similar pattern. We're starting to see a lot of A, a lot of T, and they're almost in equal quantities, and not much G and C, at least in the DNA that he pulled apart. All right. So, yes, at some low levels, there's some evidence that you could have maybe A equal G equal T equal C roughly, but most certainly for multicellular organisms, this is simply not true. All right, so this says that something's wrong with this hypothesis, right? There's really it can't be some nice little perfect ATGC structures that there's too much A and T at times. But so there's a lot of thought about this. So Chargaff, though, did propose, okay, and some ideas. And in fact, he actually met with Watson and Crick. in about 1952 and kind of told them about some of his data, which is going to become very important as we talk about the structure. But what did he see? Well, in DNA, the amount of A is approximately equal to the amount of T, okay, and G is equal to the amount of C. This is known as the concept of pairing, okay, the idea that somehow A and T are linked together potentially, and G and C are linked together, okay, but not A to G, okay, so the concept of pairing. In DS, which is we're going to use for double-stranded DNA. You'll sometimes see SS, that's single-stranded, but we're talking about double-stranded DNA. The sums of the purines equals the sums of the pyrimidines. Okay? Now, what he then found was that the amount of G and C, so this is one ratio, this is his others, the G plus C is not equal to percent of A and T. And you saw that in humans, there was far more A and T than there was G and C, and this varies across various organisms, okay? It's not a constant. All right, so what else he observed was the amount of GC actually increases what's known as thermostability, right? So if we think about this for just a moment, thermostability is based on the idea that these little hydrogen bonds that are out there control how fast we can pull apart DNA or move it, okay? Now, A to T, excuse me, has 2 bonds. G to C has 3 bonds. I need to draw another bond in there somewhere, but it has 3 bonds. Okay, and so I'll get something out here where I can see it. There we go. So this is slightly incorrect. There should be 3 little hydrogen bonds there. Okay, and so we have this idea that A to T, G to C. What that means is that things that are found in warmer areas should have more GC because you've got 3 bonds, hydrogen bonds, holding those double-stranded DNA together. When you just have A and T, you only have 2. So the prediction is, and the reality is, in regions of DNA that have more GC, because it's not constant throughout the DNA, that's more thermally stable. What it means is that if you just heat the DNA up, DNA will pop apart. Double-stranded will form single-stranded. And the areas that have more GC will be slower to come apart than areas that have AT. They'll be slightly faster to come apart. Okay? All right. So, as I said, he met with Watson and Crick, and he told them about these ideas that there was pairing between A and T and G and C, that there was not equal amounts of these, and that we have these sort of ideas going back to Levine and others about the sort of bonding that's going on. Okay. Now, here's our basic idea, the base pairing. Base pairing is the fact that if you take a purine and a pyrimidine, in this case cytosine and guanine, that you're going to have 3 hydrogen bonds that are going to form, okay? And if you pick another purine pyrimidine, but this time it's thymine adenine, you're only going to have 2. So A and T give you 2 bonds, G to C give you 3 bonds. All right? Okay. So what have we done? We've disproven this tetranucleotide hypothesis. There's not a perfect little square of ATGC going up because clearly there's not the right amount of DNA or nucleotides in DNA. So what were the alternatives? Well, Linus Pauling had the idea, a very brilliant man, okay, that it was a 3-stranded helix. And he thought it was a 3-stranded structure, okay, that formed some kind of helical form. And he came up with some basic chemistry to argue for this. But it did a weird thing, okay? It basically said that the phosphates would be in the middle, okay, somewhere in the middle of the core of all this, okay, near the axis. And if you really looked at it, he could never get it to stabilize because the van der Waals forces here were just too weak and they're too short. They need to be a certain length. They weren't going to function. So no one, even though this was a model, no one could really come up and say how this was going to work. It didn't seem to fit. Now, again, there was no real blueprint. We didn't know what it looked like, so people were just proposing models, and certainly Linus Pauling's model was one that was at least on the surface feasible. So we had 2 big models, right? We had this first tetranucleotide model that had been around for a long time. It gets thrown away. No, we don't get it. We take this individual who's a major chemist, is a Nobel laureate, comes along and says, well, we take 3 strands and put them together. They'll form a helix, and we'll end up with this nice structure that looks like this. Okay, so that's where we were. All right, we're right now in the early 1950s. We know it's DNA. That's the biomolecule of life. We know what it's not, but no one right at the moment knows what it is. So let's go explore how we go from this model to what we now know about DNA as the appropriate model.